Okay, we are again starting off with the part 2 of cerebellum. Okay, it's a very simplified one. It might not be having every content of anatomy, but I'll try to give as much as concept possible. So we need to know that this is the cortical zone of the cerebellum. What are the three layers present in the cortex? I'll tell you. Now this layer, the most superficial layer, is called as molecular layer. Okay, the one where this Purkinje cells are present, they are called as Purkinje layer. Okay, this inner zone is called as granular layer. So molecular layer has, what are the cells you are seeing here? One that you are seeing is the stellate cell. What you are seeing here is called as basket cell. Okay, you can also see in the molecular layer you have two cells. What is it? Stellate cells plus basket cells. In the granule layer, can you see two cells here? One which is in the blue color, which is going up and sending the parallel fibers like a T shape. That blue cells are called as granule cells. And you can also see something in orange, that's another G. You remember G, that is Golgi cells. So in granular layer you have two G's, that is granule cells and Golgi cells. Okay, so in molecular layer you have what? Stellate cells, basket cells, plus you can also see that these blue fibers, they are called as parallel fibers. They are nothing but the axons of which cell? Granule cell. All these constitutes which layer? Molecular layer. Purkinje layer, as the name indicates, it's just the purple color cell, the large flask-shaped cells. They are called as Purkinje cells. That is all we have in Purkinje layer. In the granular layer, what do you have? We have two Gs, Golgi cells and granule cells. So out of these 5 cells, 1, 2, 3, 4, 5 cells, only granule cells are positive neurotransmitters. Rest every cells, whatever you have, they are all negative in its response. Okay, now what you need to understand, what are the afferents reaching? The one which I have drawn in pencil, they are the two afferent fibers. So what do you mean by afferent to cerebellum? These are connections of cerebellum which we are dealing with. We have two sets of fibers in the name of afferent. One is climbing fibers, another is mossy fibers. What is it? Climbing fibers and mossy fibers. The one, can you see here? This one, which is going up and directly synapsing with the Purkinje cell, one's Purkinje cell. These fibers, these afferent fibers are said to be climbing fiber. They are coming mainly from olivary nuclear complex of medulla oblongata. From the olivary nuclear complex of medulla oblongata, these fibers go high above and they only synapse with one single Purkinje cell. They are called as climbing fibers. What is the another afferent here? Called as mossy fibers. Rest, where all do you think the afferents can come to cerebellum? Like mossy fibers could be coming from brainstem, reticular fibers, reticular system rather, cortex, from the spinal cord in the form of ventral and dorsal spinocerebellar tract, from the spinal cord, from the red nucleus, all these, and from very important called as vestibular nucleus. All these portion would be reaching to cerebellum through a fiber called as mossy fibers. Whatever you are seeing here, so many, there will be many granular cells which will go and synapse. These afferent fibers called as mossy fibers will go and synapse with granule cells. Now these granule cells through the parallel fiber will synapse with so many Purkinje cells. So the precision is one climbing fiber with one Purkinje cell, one mossy fiber with literally 500 Purkinje cells. That's a precision. 1:1 climbing. When it comes to mossy, it is 1:500 Purkinje cells. Understand? So these are the afferent which you need to know. Efferent, what have you understood about cerebellum? Efferent. Afferent, I hope it is clear. Climbing fiber from the olivary nuclear complex. Mossy fibers from the rest of the part of our CNS. Okay. Efferent, where all it can go. And you also need to understand, if you see, everything is literally having synapses with each other. At the end, these fibers will all control the action produced by Purkinje cells, and the axons, can you see it is coming down? And these axons of Purkinje cells are said to be main efferent. What is it? Main efferent is by what? Axons of Purkinje cells. That's a main efferent. Where do they go and synapse? They actually go and synapse with deep cerebellar nuclei. Deep cerebellar nuclei. You already know from the previous topic I was talking about deep cerebellar nuclei. What are the deep cerebellar nuclei that we have? From medial to lateral, we have vestigial nucleus, nucleus globosus, emboliform, and dentate nucleus. How do you remember? Dentate D, don't eat emboliform. Global globosus, food is vestigial. Don't eat global food from lateral to medial. Now these are the, now imagine this could be dentate or this could be emboliform or this could be vestigial. It depends on which lobe Purkinje cells it is, isn't it? They will go and synapse with these deep cerebellar nuclei. Deep cerebellar nuclei. Now from the deep cerebellar nuclei, they go to certain stations. That could be either red nucleus mainly. First efferent is red nucleus. Thalamus. Okay, especially ventral tire of thalamus. Yeah, ventral lateral nucleus of thalamus. Yeah, to the spinal cord, very important. Yeah, spinal cord, it will go the efferents from the cerebellum. Yeah, and to from the thalamus, it will definitely go to the cortex, cerebral cortex. Cerebral cortex. These are the efferents. Okay, red nucleus, thalamus, spinal cord, cerebral cortex at the end. So what is the ultimate efferent? They are nothing but the axons of Purkinje cells. Where do they go and relay in? They will relay in deep cerebellar nuclei. From the deep cerebellar nuclei through the cerebellar peduncle, they will be either going to red nucleus, thalamus, spinal cord, cerebral cortex, reticular fibers. Yeah, so many efferents it will send. But one thing let me tell you, that the entire cerebellum is connected to brainstem by three cerebellar peduncle, that is superior cerebellar peduncle, middle cerebellar peduncle, and inferior cerebellar peduncle. Now these peduncles will be connecting cerebellum to midbrain, pons, and medulla. Via superior cerebellar peduncle, midbrain is connected to cerebellum. Via middle cerebellar peduncle, it is connected to pons, and via inferior cerebellar peduncle, the medulla is connected to cerebellum. Via these peduncles, very important cerebellar peduncle, the afferents from these aforesaid stations will be either entering into the cerebellum or leaving from the cerebellum. But you need to understand that the main afferents, afferents, will be entering through which cerebellar peduncle? Afferents mainly will be entering through inferior cerebellar peduncle. Very important. There are cerebellar peduncles, yeah, like middle, superior, but main afferents, they enter through inferior cerebellar peduncle. Inferior cerebellar peduncle. Okay, inferior cerebellar peduncle will be sending from medulla, spinal cord. Okay, middle cerebellar peduncle, let me tell you, from the pons, like you all know, this is cortex, imagine. Okay, cortico-pontocerebellar, you remember that? Cortico-pontocerebellar. Now these fibers will be entering through, this is also very important afferent, they will be entering through another, that is middle cerebellar peduncle from the cortex. So you have understood inferior cerebellar peduncle kuda, it will be entering from medulla or the spinal cord. From the cerebral cortex, that is our main brain, it will be reaching from there via middle cerebellar peduncle. The efferent, now if you need to talk about the efferent that is from the cerebellum, like just now we spoke about, they will be leaving through mainly superior cerebellar peduncle. Very important. Superior cerebellar peduncle. They'll be going to the red nucleus, yeah, then from there to thalamus, then from there to cortex. Okay. Okay. So I have made this simple flow chart, this tabular columns for you to have a better understanding. This will be very important for you. Archicerebellum, like you remember, embryologically it's the most ancient, and it is well developed in fishes and amphibians. So why fishes and amphibians? It is very, if you see the entire three parts of a cerebellum, that is arche, paleo and neo is well developed and the largest of all in us, that is humans, primates. Arche, if you see that will be more well developed in these lower vertebrates, especially fishes and amphibians. So what is the main function? FTN, yeah, function. It is mainly to maintain the equilibrium. Yeah, that is the balance. Now these equilibrium, how exactly will you all know fishes really need to maintain their equilibrium balance, isn't it? And even the amphibians. Now through which nuclei they'll be receiving the afferent? This part of the cerebellum, they'll be receiving the afferent from mainly what? You all know balance is by vestibular nucleus. So the afferent is mainly by vestibulocerebellar tract. Understood? Vestibulocerebellar tract afferent. They will go and relay in which nuclei? Like you remember that the fastigial nucleus is the one which is in relation to archicerebellum. So we'll write here fastigial nucleus. In which zone was fastigial nucleus? Remember middle zone? Yeah, that is vermal zone. Vermal zone. In the middle part of the nucleus. Vermal zone. Okay, vermal zone. That is the vermis, literally. From there, now this fastigial nucleus will have to leave the efferent, right? Where do they leave? They will leave its efferent, yeah, through vestibulospinal tract. Vestibulospinal tract. Spinal cord, that is efferent will go to the spinal cord. Via which tract? Vestibulospinal tract. Okay, it is mainly formed by what, guys? It is mainly formed by which part of cerebellum? That is flocculonodular lobe plus lingula. Flocculonodular lobe plus lingula. Okay, lesion of this, if in case archicerebellum there is a lesion, what would happen? Our equilibrium is lost, so there will be no balance. So that how it will be presenting in a patient, how it will be manifesting, that is ataxia. Patient won't be able to stand straight. He will sway or fall into the side of lesion, to the same side of lesion. Let me tell you, cerebellum will maintain the same side of our body. It will balance the same side of our body. Yeah, so there will be ataxia. Not only that, we'll also have nystagmus, because this part of cerebellum is said to be coordinating our eyeball movement. Yeah, so nystagmus. Fine. We'll go to paleocerebellum. Similar fashion only we'll be going to paleocerebellum. What have you understood? So what is forming the paleocerebellum? Mainly you remember? Yeah, the orange color which we had discussed in part 1. That is nothing but our anterior lobe. Anterior lobe of cerebellum excluding lingula. Lingula. You just remember that, that's all. I have not spoken about it. Anterior lobe excluding lingula. So embryologically it is the next developed, ancient being arche, the next being developed is paleo. It is very well developed in birds and terrestrial animals. What do they require mainly? Birds, terrestrial animal, what do they require? What is their main function? They'll have to maintain their posture plus tone of their muscle. Isn't it? Yeah. So what do you think, who is maintaining all this? Yeah, which nuclei is responsible for receiving the afferent? By the way, before that, to that, where does it receive the afferent mainly from? Yeah, it receives afferent mainly from, what is it? Yeah, it receives afferent from spinal cord, isn't it? So we would say it as spinocerebellar tract, spinal part, spinocerebellar tract, tone, isn't it? Tone of the muscle, spinocerebellar tract. What is the nuclei responsible for this part of the cerebellum? That is our nucleus globosus plus nucleus emboliformis. Together we have a name called as nucleus interpositus. Nucleus interpositus. Which zone of cerebellum is this located? I mean, this part of cerebellum is located. That is paravermal zone. Please do refer to the first part of cerebellum so you'll have an idea. Paravermal zone. Efferent, where do they go? They, from the nucleus interpositus, they would go into red nucleus. Red nucleus. Okay. In case if there is a lesion here, what would happen? How would be it manifesting in a patient? Obviously, the tone and the posture will be lost. So the gait will be, what is the gait in cerebellar lesion? How will be the gait? Little wide based, like a drunken gait. Drunken gait, wide, the legs, lower limb will be like kept very wide apart and they'll walk without getting any balance. So that's gait will be lost. Yeah, it will be very classical feature. Okay, apart from that, tone is lost. So how will be the tone of the patient? Tone will be very very flabby. Hypotonia will be there. Hypotonia will be there, including tremors. Now tremor is very classical here. That is called as intentional tremor. Resting tremor means when the patient is not doing anything and sort of resting. That is a sign of Parkinsonism. That is a different scenario. In this tremor, if pen is kept here and you ask the patient to pick up the pen, they'll have the sort of intentional tremor. Now when they are resting, there is no tremor. When they are going to do some activity only, there will be tremor. That is called as intentional tremor. Okay, I hope you understood. Then neocerebellum, just like that. What is the main function of, where, by the way, in which embryological it is well developed? I have already told you, it is in primates. Primates, that is humans plus humans. Yeah, humans in us. So which part of the cerebellum is mainly forming neocerebellum? That is posterior lobe. Posterior lobe. Yeah, mainly the posterior lobe. What is the nuclei, from where it is receiving mainly? I mean, afferent is mainly from cortex. Cerebral cortex. Yeah. What is the nuclei in relation to neocerebellum? That is dentate nucleus. You remember we spoke about dentate nucleus. What is the efferent then? It is going to the red nucleus plus thalamus. You remember this portion as dentato-rubro-thalamic fibers. What is it? Dentato-rubro-thalamic fibers. That is cortico, I told you, cortico-dentato-rubro-thalamic fibers. Okay. Now what is the zone? It is lateral zone. That is cerebral hemisphere. The rest of the cerebral hemisphere, lateral zone. What is the function, guys, that is helping in maintaining our coordination of movements, fine movements, isn't it? If this has been lost, what will happen? We won't be able to do certain coordinating movements like this, supination, pronation. That is called as dysdiadochokinesia. What is it called? Dysdiadochokinesia. I'm sorry if the spelling is wrong. Dysdiadochokinesia. That is one thing. Yeah, we won't be able to touch the examiner's finger with our nose. That is called as finger nose test. What will happen? We'll pass the point. Okay, starting will be difficult. We won't be reaching our nose. We'll go poke our eyes, poke our cheeks. That is called as dysdiadochokinesia. Okay, all these together is called a cerebellar lesion. I hope you have understood little of it. Thank you so much.